Good afternoon, my name is Yobana Marino Cardenas. I am a postdoctoral researcher at the University of Puerto Rico, and I have been working with the coffee berry borer since 2010. And today I'm gonna talk about the life table and perturbation analysis of the coffee berry borer and their implication for management of this pest. The results from this research has been published in the journal PLOS ONE, in the case that you want to see the paper. As introduction, we have that the coffee berry borer, or Hypotenemus hampi, is the most important coffee pest worldwide. The management of the coffee berry borer is difficult because the entire life cycle occurs well protected inside the fruit. Only when the adult females are out searching for new fruits to colonize is the appropriate moment to apply a control measure like biological or chemical. This is the position A and position B. And this period is short period of time. And also this behavior makes it difficult to study the biology and the development and the life cycle of the CBB in the field. One strategy that could help us to improve the management of the coffee berry borer and could help us to study the coffee berry borer in the field could be a life table analysis, because these type of analysis are important tools that let us to know important biological parameters of the insects, for example, the coffee berry borer at individual level and the population level. On individual level, we can determine important parameters like egg viability, fecundity, mortality, and developmental time. And at the population level, we can determine important, we can calculate demographic parameters like population growth rate. The population growth rate value indicates the status of the population. If the value of the population growth rate is more than 1, this means that the population is increasing in size over the time. I will explain in more detail what means these other demographic parameters in the results section. Life tables also are the base to perform a demographic analysis. These type of analysis are important tools that let us to know about the population biology and the other different characteristics of the population. And this type of analysis could be used in two ways. For example, to protect endangered species or to improve the management of insects pests like the coffee berry borer. This type of analysis of demographic models are developed from projection matrix. Here is an example of projection matrix. Here we have the population at the beginning of the evaluation. For example, in the case of the coffee berry borer, we have a population of eggs. And here is the population and the end of the evaluation. For example, after 56 days of evaluation, we have a population that could be formed by eggs, larvae, pupae, adults, and different stages of development of the coffee berry borer. And we use this data to construct the projection matrix. The projection matrix contains the fertility, in the case of the coffee berry borer, fertility of adult females, and contains different probabilities, mainly probabilities to survive and move into the next stages, or probabilities to survive and remains in the same stage. I will explain in more detail a projection matrix in the results section. One important demographic model that could help us to improve the management of the coffee berry borer could be those called prospective perturbation analysis. Because this type of analysis predicts before this happens the effect of on population growth rate when the population is impacted or the population is growing in the different conditions. For example, the coffee berry borer growing on the sun under shade, or population impact when the population is impacted for natural enemies like ants or like the fungus Baueria bassiana. This type of models are based on the values of sensitivity. Sensitivity values indicates the contribution of the different life stages or life cycle transition of the coffee berry borer to the value of the population growth rate. For example, one life cycle transition of the coffee berry borer could be the probability to survives as egg and moves into the larva. And transitions or life cycle stages with higher values of sensitivity could be important targets for the management of the coffee berry borer. Our objectives were to construct life tables from population of the CBB growing under field conditions and identify life cycle transition of the coffee berry borer with higher values of sensitivities. And finally, to conduct a perturbation analysis. The objective of the perturbation analysis is to find the point at which the value of population growth rate is less than 1. This means that the population is decreasing over the time. And to conduct this perturbation analysis, we only modify or reduce the values of the probabilities of its life cycle transition until the point that, as mentioned before, the value of population growth rate is less than 1. We made artificial infestation in the field. For this, we use CBB adult females from the artificial diet Cenibroca. And these females were disinfected and put into the Petri dishes with the filter paper. And these females were taken the same day and were taken to the field. And in the field, we selected different coffee branches. And we target these branches and we manually remove all the fruits that were perforated by the CBB. After that, we put groups of 50 females into the clear plastic bags and we tied these bags to the branches, and 2 days later, after we put the adult females, we remove the tags, the plastic bags, and we tag all the fruits that were perforated by the coffee berry borer adult females, and we tag these fruits with nail polish. We did 3 artificial infestation from August to September 2018. And we use a different color of nail polish for each infestation. For laboratory evaluation, we collected weekly from 7 to 56 days after infestation, 50 infested fruits, and we take these fruits to the lab, and with the help of the stethoscope, we counted all individuals inside each fruit. We counted the number of eggs, larvae, pupae, juveniles, and others. And also we determined the presence, survival, and positive oviposition of the adult females. Positive oviposition means a positive reproduction or presence of different individuals inside the fruit. To perform prospective perturbation analysis, we divide the life cycle of the coffee berry borer in 5 stages. We divide in eggs, larvae, pupae, juveniles, and adults. We use the developmental times that we determine in lab experiments because in field it's difficult to follow the same organisms in the time. So we use these times that corresponds to population that were growing at the lab and 25 grade Celsius. For example, the time of developmental time from egg to larvae was 7 days. From larvae to pupae, that this time includes the 2 instant, larvae 1 and larvae 2, and the pre-pupae stage was 17 days. From pupae to juveniles were 7 days, and from juveniles to adults were 4 days. We use these times to calculate the different probabilities. To calculate the probabilities GA, that was the probability of transition between stages in which an individual survives and moves on the next life stage. And for example, we count the number of larvae that were present at 14 days after infestation and the number of eggs that were present at 7 days after infestation. We use these values to calculate the value of GA. We divide the number of eggs in the number of larvae, and we found the value of GA, or probability to survive as egg and move into the larvae. And we made the same for the other stages. Also, we calculate the probability to survive, probability PA, or probability to survive and remains on the same stage. For example, here is PA, that is the probability to survives as egg and moves in and remains as egg. And here is the P5 is the survival of the adult females. And in the case of the coffee berry borer, only the adult females contribute to the population growth. So we only calculate the fertility of the adult females. And here is our projection matrix. Here, as I mentioned before, we have the fertility of adult females, the probabilities PA, or probabilities to survive, remains in the same stage, and GA, or probabilities to survive and move to the next stage. And here is our projection matrix with our data from field. For example, here we have the probability to survive as egg and move into the larvae. This was 0.89. This means that the 90% of the eggs survive and move into the larvae. And here we have the probability to survive as larvae and move into the pupae. And here is the probability, the probability to survive as pupae and move into the juvenile. And this probability was significantly low. Only 39% of the pupae survive and move into the larvae. Here is the survival of adult females. In our case, was 80% of the adult females survive. To conduct a prospective perturbation analysis, as I mentioned before, the objective of perturbation analysis is to find the point in which the value of population growth rate is less than 1. So for do this, we reduce the values of our probabilities in our projection matrix to the point in which the value is, the population growth rate is less than 1, as I mentioned before. We conducted 2 types of analysis. First, we modify all life cycle transition at the time. We modify only one cycle transition and the other transition remains constant. Second, we modify the transitions of the life cycle transition or life cycle stages with higher sensitivities. In this case, these stages or these values of probabilities were reduced at the same time. As results, we have that for a population of the coffee berry borer growing under field condition on the shade coffee plantation of Coffea arabica with mean temperature of 21.8 Celsius grade Celsius, the population growth rate was over 1, was 1.31. This means that the population is increasing in size over the time. We also calculate the mean generation time, that is the developmental time from egg to adult, and this was 40 days. We also calculate net reproductive value. This was 20.3. This means that one female produces 20 females for each generation. And we also calculate the double time. That was 13 days. This is the time required to double the population size in the time. For example, to pass from 50 individuals to 100 individuals, in the case of this population, the time is 13 days. Here is the results from sensitivity analysis. In the y-axis are the values of sensitivity or the value of the contribution of each life stages of the CBB or life transition of the CBB to the population growth rate. And here are the different probabilities. GA are the probabilities to survive and move into the next stage. And we observe that these probabilities were higher than the probabilities PA that were the probabilities to survive and remains on the same stage. And here is P5, that is the survival of adult females. Here we observe 3 probabilities or 3 life cycle transition with higher values of sensitivities. These were G2 or probability to survive as larvae and move into the pupae, G3 or probability to survive as pupae and move into the juvenile, and P5 or survival of adult females. These 3 life cycle transition could be important targets for the management of the coffee berry borer. The results from perturbation analysis showed when here we have in the y-axis the values of the population growth rate. Here is 1 and here is our value of population growth rate. And here are the different probabilities to survive and move between stages. And we observed that when we modify only, or we reduce only the value of the probability of only one transition, the percentage of change are higher. We have to reduce, for example, the survival of eggs to 80% to 10% to reduce the value of population growth rate to less than 1. And here, in the case of the survival of adult females, we reduce the value of survival of the adult female from 80 to 0, and the population growth rate value continue be more than 1. But when we reduce or modify the values of the probabilities of the 3 life cycle transitions with higher sensitivities, at the same time, we'd observe that the chain necessary to down the value of population growth rate to less than 1 is reduced. For example, we only have to reduce to 0.35% the survival of larvae, to 50% the survival of pupae, and 55% the survival of adult females. And here in this point, the value of population growth rate starts to decrease. As conclusion, we have that the survival of adult females, survival of larvae and pupae could be 3 important targets for the management of the coffee berry borer, because these 3 life cycle transition or life stages had higher sensitivities. Also, we observed that an efficient management of the coffee berry borer should not be focused only in one target or developmental stage, because a higher levels of change or impact has to be done to reduce the value of population growth rate. Integrated pest management programs of the for the coffee berry borer should be focused to enhance the diversity of natural enemies like ants, parasitoids, and predators, because these organisms could have a great impact on the survival of larvae, pupae, and others. Other measures like biological measure controls like the fungus Beauveria bassiana, and cultural measures like sanitation, that is the collection of all fruits that remains in the branches after the main harvest, are great tools to reduce the survival of adult females. This research was funded by USDA ARS and Puerto Rico Science, Technology and Research Trust. And we thanks to the Dr. Luz Cerrato and Rocio Rivera for their help in the field and the laboratory. And thanks for your attention. And if you have any question, I'm here and ready to answer this. Thank you.